Okay. Good morning. So I'm going to continue on with the topic that Cheryl started. Cheryl's a tough act to follow. I'm going to try to focus more specifically on reproduction in fishes and our efforts to model that in salmonids, and particularly female rainbow trout. And I thought we'd start with just some general thoughts. A lot of this Cheryl's covered, so I'll go through it a little quickly. But so the obvious thing is when you're trying to develop these models across multiple scales, it's hard. But an aspect that was pointed out in one of Cheryl's slides is the time scales that you deal with can change dramatically or abruptly. And so that can influence how you build the models and how they connect together, particularly at the lowest level where many components at the cell subcellular level operate quite fast. And so you can get dramatic changes in some parameters at that level that may or may not be reflected immediately upstream. And so models need to be able to handle that or explain it. Also, when you seek to develop these models, and that maybe it starts to build on the question that was asked a moment ago, is where do you start? How do you want to parameterize these things? And among fishes, there's just, aside from the obvious great diversity in types, there's great diversity in data sets that are available. And some fishes, particularly these small aquarium fish models, zebrafish, but fathead minnows too, it's kind of interesting in that you have what's emerging is rich data sets at this level, gene expression, some proteomic data, and then relatively less sort of at the middle level. But then when you get to the whole animal level, there's these nice data sets emerging with behavior and reproduction. And so some of the models that are coming out are sort of trying to connect those two levels. Other fish like rainbow trout that are sometimes thought of as the rat of the aquatic world, that there's quite a bit of knowledge known about trout physiology sprinkled out across all these levels. And so it gives you a little bit of a different perspective about where you want to begin. And so the models that we're working with probably might be viewed more as a top-down or middle-out approach because of the relative wealth of data across different scales. But also when we're thinking about how we want to take advantage of other people's work and connect with our models, particularly at some of these biochemical network models that are emerging from the gene expression data, there's greater diversity in how people are choosing to build models, and we found it challenging at times to connect what we're doing with some of the outputs coming from some of these other types of models. And so that's still a challenge to work out. And then and the last thought is when you seek to add greater complexity to your model, inevitably you face a point where there are just gaps in knowledge of physiology, and you're faced with this question, then how do you handle that? Do you want to continue to add complexity and add parameters to the model and then estimate parameter values for those, or do you want to pull back and take a more empirical approach to leap over these gaps in knowledge? And it seems to me that's still sort of done at the individual modeler's level, those choices. There's not a lot of consensus about when to make those decisions. And Cheryl's done a nice job sort of going over the adverse outcome pathway and where models are expected to fill in. And I just would add a couple of thoughts. Just recently I was at the Society of Toxicology meeting, and so within the human tox area, they've changed their nomenclature, so now they're using AOP also. So now the term is encompassing both human and eco. And so there's a lot of use of it, but yet it's just striking how little modeling is actually involved even there. A lot of people with their in vitro or high throughput data that they're collecting, they're taking that immediately and assuming that it's an equivalent whole animal response and then going from there. So they still have a ways to go at incorporating models into their work. But I think, as Cheryl pointed out with the National Academy document and this plan or that was put forth for toxicity testing in the 21st century, the long-term goal was the continued development of these biological or physiologically based models, get them at a sufficient level of complexity that they become valuable tools to translate in vitro results to whole animal effects. And so the work that we're doing with trout in our reproductive model is kind of geared towards that last point. And just to step back a moment, when I started to get interested in this and we were beginning to review the literature maybe 10, 12 years ago, one thing that we're immediately aware is that there was a history of work by others dealing with the same problem, wanting to extrapolate from molecular level data to whole organism effects. And this was in this clinical pharmacology arena where for decades with pharmaceutical agents and prediction of human health benefits from taking or reversal of disease outcomes associated with drug therapy, there was a large body of work trying to develop mechanistic models to explain this. And one group that we really admired, thought their work really stood out, was by William Jusko. And his lab was at the University of New York, and he's mostly retired, my understanding is now. But his lab for many years put out a series of papers with different compounds, but really the work with corticosteroids was particularly valuable to us as sort of a blueprint about how you might go about in a stepwise manner developing these models, because they dealt with a lot of the issues we're faced with knowledge gaps that later were filled in, and so they could go in and revise their models over time. And so I suggested this paper for reading, and this was a paper that he wrote last year. It's kind of an introduction and brief review to some of what they were doing in the context of this larger area in clinical pharmacology. And these were some thought points that he mentioned in the paper. And I like them because it sort of reflects a little bit the approach that we've tried to use in our model development with reproduction in fishes. And so he makes the distinction in the extremes in terms of types of models that have been used, what he calls basic mechanistic pharmacokinetic, pharmacodynamic models. Really, there's not a big distinction between toxicokinetic and toxicodynamic models. It's just whether it's a drug or a toxicant and an adverse or beneficial effect. But one side of the models that have a longer history are where they're relatively simple models, developed against experimental data that might be collected for a specific purpose with the models. The models then are usually rigorously fitted, usually nonlinear regression methods, and it's expected that the parameters have a strong statistical reliability as a part of the regression process. And then in the better models, then it's clear that the parameter estimates you get from this exercise have a direct correlation to some underlying physiological process that might be influencing either the dosimetry, the kinetic behavior of the molecule, or responses going on in the organism. And then on the other extreme is this so-called systems models, where the models are developed initially from a different perspective. The goal then is to identify all possible processes and agents involved in either the physiological processes in question or organismal response. Typically then this leads to large numbers of equations to describe these processes, in some cases hundreds of equations for some of the models that are out there, and nearly 1,000 parameters as part of the model. And with so many equations and parameters, it's simply impossible to have enough experimental data to accurately provide information on how to parameterize these models. So inevitably then assumptions are being made, guesswork's being made for different parameters. And the fallout from this in a lot of cases is the models are so complex, it's difficult to fully understand some of the key assumptions that are being made in the model, or at least they're hidden and difficult to tease out. It also makes it hard then to explain the models to others. And if you've ever been a reviewer on these kinds of models, it's hard in even a relatively couple of weeks time period to understand them sufficiently well to be critical of them, or it provides some constructive criticism. So that's sort of a wide swath. What we've tried to do is seek some middle ground in our development of our model and make them complex, but not quite as complex as you could be if you started, say, with a model like this. And so, Dan Villeneuve and colleagues put out this paper, and they really done a good service. So, but I used it to juxtapose against what we're trying to do. And so what Dan and his colleagues have done is they've gone through the literature and the fish reproductive literature and pulled out studies that have indicated different genes, proteins, different processes that have a role in fish reproduction, and organized it in this graphical systems model. Now this model is not coded with mathematical equations, so it's not a dynamic model in that sense, but it's a nice, very detailed conceptual model. And so if you want to do one of these systems models that captures everything that is known about fish reproduction, this is where you might start. And our approach, as I mentioned, to try to seek some middle ground, is not to focus so much on what's in these boxes and capture every one of these steps, but to focus on the transitions, that is, what is going on in the brain or the hypothalamus in terms of its key outputs, like gonadotropin-releasing hormone and its action on the pituitary, and focus more on the production of the gonadotropins and their release from the pituitary and so on. And then the idea then is to have And this sort of follows on William Jusko's group. What they had done in the past is you start out with a relatively simple model, and then you add complexity iteratively over the years based on the needs that people that might be using the model, and as the understanding of these different processes and their connection to some of the key functions going on in the tissues becomes better established. So this is our conceptual model that we work off of. So we take a big step back from that. And so then it just follows what I just mentioned. We're kind of just focusing on what I like to call the essential parts of the model. If you're starting out to build one of these models, what do you have to consider that would be universally agreed among endocrinologists? And so, as I mentioned, GnRH production by the hypothalamus and the two key gonadotropins from the pituitary. FSH in particular in trout is released continuously during the year. It has, as I'll show later, kind of a complex profile over a reproductive cycle. But among its key things it does is stimulates ovarian growth and the production of estrogen, which in turn drives vitellogenesis. And there's other things that are going on that you need to consider, and I'll touch on in a moment. But some years ago, we had a chance to do a large study to collect some data that would help inform the model and parameterize it, and just wanted to go over briefly aspects of what we did. So we worked with a local commercial trout hatchery that was an egg production facility. So they had large numbers of female trout that were spawning at different times of the year. So it helps get around this limitation of annual spawners, and that you don't have to wait till the same time every year to get your eggs or your fish. So they had females that were ripening at different times of the year. Another thing is it had large numbers of fish, so it was possible to get a relatively large cohort of females that had ovulated within a 24-hour period. And that's kind of hard to do with trout. But so we've got these fish, and we brought them up to our lab in Squim, and basically sampled groups of fish at various times over a reproductive cycle. So as I mentioned, these trout had just finished their first reproductive cycle, so they were known to have successfully spawned. And then we tracked them during their second cycle, and to measure different things related to reproduction. Another thing that we did, we were interested in gene expression changes that were occurring in some of these tissues that were important in the model, and there were a couple of goals with this study. One was just to try to get a more general view of kind of what's going on in these tissues over a reproductive cycle. Another goal, which has been a little bit more challenging than we expected to work into the model, was to try to tease out profiles of selected genes in some of these tissues that we could use the model then to characterize, and see then if we could add more to the model that to other genes and proteins that maybe previously hadn't been thought of to play an important role in reproduction. And when we started this, we were using a high-density gene array. This was a 16K gene array that was being made at the University of Victoria by the GRASP consortium. At the time, this was kind of the best tool out there to do this kind of work. Nowadays, people are moving away from using gene arrays and doing high throughput sequencing. But I just wanted to digress a moment and show a summary of some of the gene expression in work and sort of what we were able to take, but also used it as kind of an example of it may seem easier to do when you get into these studies and some of the difficulties we faced. So these three heat maps just attempt to summarize all of the gene expression changes we could measure with this array at these different time points through the cycle. And so one of the challenges was when to sample the fish. This was kind of expensive to do. We couldn't just sample as much as we would like, so we made some decisions about when to start sampling. So in the case of the liver and the pituitary, we waited to nearly 2 months into their cycle to begin, thinking that things were more in a refractory state coming out of the previous reproductive cycle. But in the case of the liver, what's interesting is almost immediately there were large changes occurring. This lowercase showing large number of genes that were immediately induced. Some of these are associated with vitellogenin, which is a little bit faded here, but there was a large number of genes sort of in this pale green that were downregulated initially. Many of these were associated with albumin and ovalbumin and other proteins in that family. Sort of reflects what appears to be a shift in the liver of preparing to make a lot of vitellogenin and moving away from other protein synthesis. Also, some more subtle things that can be teased out of this is changes in energy usage by the liver, shifts in carbohydrate metabolism, and others that appear to be associated with the tissue getting ready to ramp up this large amount of vitellogenin it's going to make later in the cycle. In the ovary, is interesting, is the bulk of the more substantial gene expression changes occur in the second half of the cycle, but it's a little bit less clear about what you might tease out about general thoughts about the types of processes that are being affected. And then in the pituitary, with this type of analyses, it wasn't clear about trends in overall gene expression during the cycle. And so we had a fair amount of time to analyze this data, and we tried different approaches. Some of it was published in ET&C a few years ago, where we would, for example, you could take profiles of estrogen during the reproductive cycle and look for cohorts of genes that appeared to be tracking changes in estrogen levels during the cycle. Did also that with the gonadotropins. And you can learn some interesting things, but in terms of doing it in a way that can make connections to the model, frankly, we've struggled. And so this is an area still as a problem. And I just mention it to go back to some of these high-density data sets that are coming out on gene expression. We still find it challenging to integrate that into the model. And so in terms of the rest of the talk and some of the other data I'm showing for gene expression, we've just gone back to more traditional PCR-based methods using for genes that we know have a direct relationship to some of these proteins or hormones that are being synthesized. So go back to our conceptual model and then our formulation of the model. This was a figure that Valerie showed yesterday. And so just to briefly touch some highlights of it. So in the pituitary, how we're handling the synthesis of, again, of the two main gonadotropins, FSH and LH. We've got parameters and equations to describe their gene expression. In the case of LH, what you need to account for is it's produced but not immediately released by the pituitary. It's stored in the pituitary during the cycle and waiting for the proper triggers late in the cycle to be released. This is usually after the peak in estrogen levels occur and begin to decline. That removes some feedback inhibition on its release from the pituitary, and then you get the surge in LH, and it triggers a number of things late in the cycle that lead to spawning. A number of the hormones, and in the case of vitellogenin that's being made in the liver, are secreted into the blood, and so we also have various pharmacokinetic parameters to describe the transport of these hormones in vitellogenin after they're released into the bloodstream. To go in a little bit more detail and split things up so it's a little bit more easier to describe, this is the upstream part of the model showing the hypothalamus, which is off the slide, and then the pituitary and the ovary. And so this set of stacked differential equations describes these processes, and here's the list of the parameters, which you realize it's a busy slide. But a couple of points that I wanted to make is this work now is currently being done on a project in conjunction with the development of in vitro assays. And so we're working with some colleagues at the University of Washington, Graham Young, and Penny Swanson at NOAA Fisheries, and Jim Nagler at the University of Idaho. And we're developing assays of isolated pituitary cells, ovarian follicles, and liver cells, hepatocytes, to characterize some of these parameters that are in the model. And some examples of these are shown in the red boxes, for instance, relating to the rates of expression of the FSH gene, its protein synthesis and secretion. These things can be measured in vitro. And so we're working on then developing assays that can characterize that in vitro and use to parameterize the model, and then can go back and then do toxicant exposures to the cells and ask questions about how does the chemical exposure affect these parameters that are being estimated. And so then it's a way to do these in vitro tests with the cell assay system and get data that's more amenable to incorporation into the model. Other parameters in the model we felt were important were measured by separate experiments, more focused, whole animal studies. And this, for example, are pharmacokinetic parameters that relate to estrogen. Estrogen production is really central to the model, so we felt it was important to have as accurate estimates as we could obtain. And so this was done in some separate experiments, and just have a slide to summarize that. And so this is an example of sort of a more focused kinetic experiment. In that slide that showed from the Jusko paper, you might consider a mechanistic toxicokinetic study. And so what we've done here is I had a group of female trout. I've actually done this with fish at different reproductive stages. For the data we published last year, this was in fish that were about the midpoint of their reproductive cycle. And so I fitted them with the dorsal aorta catheter, and then once they've been catheterized, they're housed individually in these tanks, and there's some close-up pictures. And so we've got a piece of Teflon tubing that's going in the dorsal aorta. We run it out the snout, and it's hooked up to a syringe, which floats. And if you haven't seen this before, then it's a nice way to introduce chemicals into the bloodstream and sample small amounts of blood with minimal stress to the fish during the experiment. It's also a good way to involve undergraduate students that come through the lab, so get them to help out with the sampling. And so in this particular graph, then I had 4 of these fish that were catheterized, and I gave them a bolus injection of deuterium-labeled estradiol, so we can measure it without being confounded by the natural estrogen that's in the fish. And then we get these profiles. This is an average just for display, but we take each individual profile and then do a pharmacokinetic analysis on it. In this particular study, I actually decided to use non-compartmental or statistical moment methods, which is a simpler way to analyze the data, in part because really what the purpose was to get these parameter estimates of volume and distribution in total body clearance so that we can put back into the model. And then another part of it that I'm not showing, but as I mentioned, we did this with fish at different reproductive stages. There was some question, particularly with clearance, may change during a reproductive cycle, and the use of a constant value for that in the model could be a problem. You may need to consider it changing. But actually, this value has held up over a number of experiments. So we've taken these values and put them back into the model. And then with other estimates that we got from the literature, we then ran these initial set of simulations that, for the most part, looked pretty good. We did some minor tweaking after we got the model up and running, but I'll just walk through this examples. So here is a graph showing measured mean values for FSH. What we're actually measuring is the beta subunit. What I didn't mention is gonadotropins are heterodimer proteins. They have a common alpha subunit, but each gonadotropin has a distinct beta subunit. And so for the gene expression, that's normally what you focus on. And so what we're doing then is we've got fish that were sampled immediately after their spawn. We're calling the first spawn. And then at various times then we're sampling groups of fish as they approach their second spawning event. As I mentioned, we had 140 fish. We didn't kill them all during the cycle, so we kept a group of around 20 to complete the cycle, and they all spawn between day 340 and day 350. So by this point, you're at the point when all the fish then, remaining fish are ovulating. And so expression of this FSH gene, it's bouncing around along the cycle. What's kind of interesting is that these squares here are the individual observed data in the fish, and there's some fish that are kind of doing wacky things at the beginning, at the end, but for the most part, there's nice reproducibility in the levels of FSH, and it kind of has this complex profile that tracks FSH gene expression. So this is one example where we benefit that there appears to be a tight coupling with changes in FSH gene expression in the pituitary and then corresponding circulating levels of FSH. So there isn't a big time delay in this example, and it so allows us then in the solid line as the model predicted levels of circulating FSH to do a reasonably accurate description of the observed data based on the gene expression that was measured in the pituitary. And so a couple of the consequences of FSH is it stimulates ovarian growth, shown here as relative growth of the ovary, and then production of estrogen during the reproductive cycle. And you note for most of the cycle, circulating levels of estrogen are quite low in the fish, and then they begin this initial slow rise and then spike towards the end of the cycle. So just going to shift to talk a little bit about vitellogenesis. It's the other part of the model. And then I want to use this as an example of what didn't quite work out well initially, and it caused us to go back and kind of rethink some things about the model. And so this is just a simple overview of the process of vitellogenesis. So it is driven by estrogen. Estrogen is 98 to 99% bound in plasma. There's specific proteins that are made to transport estrogen. It's assumed the unbound fraction is what diffuses into the hepatocyte, binds to its estrogen receptor. There are 4 in trout, 2 subtypes, alpha and beta, and then each one has an alpha 1, alpha 2, beta 1, and beta 2. We actually think it's the alpha 1 that does most of the vitellogenesis, but binds to its receptor, triggers the expression of the vitellogenin gene and protein secretion into the plasma, and then it is assumed in actively growing female fish that the loss of vitellogenin from plasma is being driven by this rapid uptake into the oocytes. So this is then sort of our mathematical description of these processes. Another conceptual view of it. I just would take a moment to explain that we've also added this extra compartment of distribution of vitellogenin into other non-ovarian tissues, and that is one area we found was important to maintain mass balance for the vitellogenin, and I'll go into that a little bit more in a moment, why that is. But these are the set of stacked equations to describe it. There's 8 equations we're using to describe this process and the corresponding set of parameters for it. Like I showed a moment ago, in red are parameters that we're currently measuring or trying to measure with in vitro methods in cultured hepatocytes. And the focus here is on freshly prepared hepatocytes, but hepatocytes also are amenable to cryopreservation, and recognize that for high throughput screening, people aren't always going to have access to adult trout to make this. Also, the reproductive status of the fish could influence the response of the hepatocytes. So research focus right now is, one, developing ways to use the hepatocytes and get in vitro estimates of these parameters with and without chemical exposure, and then also verifying that you can cryopreserve the hepatocytes and then pull them out of the freezer later and repeat these kinds of experiments to make it more amenable to more labs getting involved in this type of testing. And then these parameters here are things that we measured in vivo in special experiments, analogous to what I showed a moment ago for estrogen. But these experiments then were trying to characterize in more detail the pharmacokinetic behavior of vitellogenin after its secretion in plasma. So what I would start then is showing what didn't quite work. So we formulated the model, went through the process analogous to what I described for the pituitary and ovarian portion, made some adjustments to the parameters and ran simulations, and this is kind of what we initially got. In terms of describing the alpha-1 estrogen receptor mRNA, I mean, it was okay. You could quibble that it could be improvements made there, but this we felt could probably have been dealt with, again, more fine-tuning of the model. The bigger problem was shown on the right. So up here, looking at model predicted values in the solid line, and these were observed data for the vitellogenin gene expression in the liver. And it's clear that the model predictions are late. It's a delayed rise and a delayed peak relative to what the observed data said. And here, if this was the only problem, again, you might say you could deal that with sort of adjustments to the model. But the other problem was that the prediction of circulating levels of vitellogenin in the blood, in blood plasma, also had a problem, but it was kind of going the opposite direction. The model was predicting a premature rise in vitellogenin, and it was peaking early. So these kind of were going opposite to each other. We recognized any changes we made to fix this was going to make this worse. So it kind of caused us to rethink the model structure and where we might need to make improvements. We also saw this as sort of this balance between synthesis and loss. So we're predicting circulating levels. The loss of vitellogenin from plasma is, again, assumed to be this rapid uptake by the ovaries. There is some nice data in the literature from 20 years ago by Tyler and Sumter and Rainbow Trout where they were able to label some vitellogenin, radio-label some vitellogenin, inject it into trout. And so a lot of us used their data to initially parameterize this process. But the question then is, should we introduce some changes here? Let's perhaps create some added delays between expression of the gene and synthesis of the protein and then its secretion to artificially introduce some time delays to try to improve this upper fit without making this situation worse. And the feeling was that that's not biologically accurate. And some data that we had collected years before kind of would support that. And I'll just take a moment to show an example here. This was a study we published in 2001. This was in male trout, naive to any prior estrogen exposure. And what I did here was, again, catheterize these fish, analogous to what I showed in an earlier slide. And I gave them bolus injections of this very potent synthetic estrogen, ethionine estradiol. And other work that I had done, it was clear that EE2 has very rapid kinetics. When you give these bolus injections, within minutes you're activating estrogen receptor. So that's a very quick step. And so one of the questions we were interested in is then how long does it take to make the vitellogenin in an intact fish and secrete it out of the liver? And so if you just sort of focus on that little bit there and blow it up, that's what's shown here. And so we were better prepared in this study. We sampled intensively early on after injection. And so what's interesting is the time delay is about 8 hours, 8 to 12 hours before you see this increase in circulating levels of vitilogenin. So that sort of gives you an idea of how long it takes between receptor activation and secretion of synthesized protein. The other interesting thing is we did this at a range of doses from a microgram per kilogram up to 10 milligrams, so a 10,000-fold range of administered doses, and this time delay was essentially constant across all doses. So I always felt that was nice, that's at least one assumption we can make, and it was supported. And it also suggested that problems with those simulations lie beyond the time it takes to make the protein and secrete it. So our attention then shifted more towards the clearance of vitilogenin from plasma and processes that are controlling its loss. And as part of that, we also took advantage of some data we collected in that earlier study. And this was a study to better characterize the vitilogenin pharmacokinetics in fish that don't have an ovary. So these are, again, naive male trout. So there's background levels of vitilogenin are very low. We had some purified trout vitilogenin, injected them again intravascularly in several fish, and then characterized its pharmacokinetic behavior. In this case, it did choose to fit the data to a compartmental model, in part because we recognize there's a number of processes going on. There is some distribution into non-reproductive tissues, and this allowed us to characterize the kinetics of that behavior and put it back into the model. Also from this study, we realized this sort of non-ovarian uptake of vitilogenin, that is, the clearance processes outside of sequestration by the ovary, are quite slow. So this value of clearance, 2 liters per hour per kilo, is about 1% of the value that we've estimated from the data of Tyler and Sumter in terms of the rate of vitilogenin uptake or removal from the plasma by actively growing ovaries. And so this suggested this is sort of this minimal baseline value for vitilogenin clearance in the absence of active uptake by the ovary. And then it got us thinking that this assumption we had made in the model, having a constant value for vitilogenin clearance based on absorption into the ovaries, was probably not accurate. And there's some other ancillary data to support that, some of which I try to summarize in this slide. So this was work done by Group Perazzolo, published in 1999. I think they were the first. There's additional work done to kind of support this. And what they're measuring is the transcript levels of the gene coding for the vitilogenin uptake receptor. And so the theory about how vitilogenin gets incorporated into the oocytes is there's a specific membrane-bound receptor that it binds to, and then through a process of endocytosis, it gets into the cell. And I'll show in a moment that there's different fates of what can happen to that receptor once it's been internalized. But what's interesting, what they had observed was the gene expression for that receptor occurs early in oocyte development. So this x-axis is reflecting the diameter of ovaries or different oocytes in the ovary, and then we're looking at the relative expression of the gene for this receptor. And the bulk of the expression occurs when the oocytes are relatively small or in an early state. And so it leads to this hypothesis that I think has been supported by other evidence that the bulk of the receptor production occurs early in oocyte maturation, and then it's shut down. And so the oocyte is reusing this receptor after it's made as needed as vitilogenin levels rise. And then at some point late in the cycle, when the oocytes become fully mature, they stop growing and they lose this capacity to absorb vitilogenin. And we're working with this little conceptual model that tries to capture some of this, and we're trying to currently update the ovarian portion of the model to incorporate these steps. And this tries to summarize that, where you've got a available receptor that can bind vitilogenin, it's internalized into the egg, then the receptor has a couple of fates that can be immediately recycled to the surface of the membrane to pull in more vitilogenin, or it can exist in these 2 internalized states, still intact, waiting to be recycled, or degraded. And so the assumptions that we're now working with is over time, as the eggs develop and approach their maximal size, most of the vitilogenin receptors are degraded, and so then the eggs lose this capacity to absorb vitilogenin. So then we tried this very simple approach to adjust our model, where this is what we originally assumed was this constant uptake clearance for vitilogenin from the plasma being driven by uptake into the oocytes. So we've kept this really simple assumption, and then when the eggs reach a certain size based on what we think is their maximum diameter within the fish. Then we transition down to this minimal value that was determined from these earlier studies in the male trout. And so then my last slide here, this summarizes the revised predictions we made. We've made some other tweaking to the parameters to get these simulations, but the really big thing was the shift in vitilogenin clearance from plasma. And so it had a significant effect. So now this line is reflecting the mRNA levels of the gene, and so through some tweaking of the parameters, we better describe this early rise in expression of the vitilogenin gene, but adding this feature of clearance from the plasma then provides a much better fit to circulating levels of vitilogenin. And of course, the model doesn't prove biology. It just suggests this is an area that we want to focus on more. And as I mentioned with that conceptual model, that's another proposal that we have out to try to go back and do some more experimental work to try to more definitively establish this change in availability of vitilogenin receptors on the oocyte. So just want to acknowledge, I've used we a lot in this talk, so there are lots of people that have helped with this. Just really need to mention Penny Swanson, Jim Nagle, and Graham Young. They're my collaborators on this project. It's trying to do the in vitro testing and incorporate it into the model, and that's funded by EPA. And then for many years, we've also had NSF support to develop this, and Bill Hayden, Yang-Han Kim have been very instrumental in that, and Sharon Hooks helped out with the gene expression data. She's now in Australia. And then, as I mentioned, Trout Lodge is a great resource because they're the world's largest producer of trout eggs for sale. And so they have a staggering amount of female fish. They're continually breeding and spawning. And so they've been very generous to give us fish when we need them. And then lastly, we have a session on a related topic at CTAC. So I wanted to add it to Valerie's list, shaping up to be a big meeting. But John Nichols and I are trying to organize a session on quantitative AOP models and in vitro, in vivo scaling. So you can make Vancouver.